concentration of sodium in the body and sodium in the ocean is the same. The concentration of magnesium is the same. The concentration of all the other salts is the same. Then it is an ionoconformer. Okay, it doesn't have to use any sort of energy to balance the water concentration, balance the salt concentration. However, some of these marine animals, even those that have a long evolutionary history in the marine habitat, might still be isosmotic for their total solute concentration, but the ionic composition is different. It might have higher sodium concentration than what's in the ocean water, but lower magnesium concentration than what's in the ocean water. But the total concentration of all the solutes taken together is isosmotic. There's no net movement of water. However, they have to expend energy to maintain a different ion constitution than what's in the marine water. Okay? Ionoconformers, they don't have to expend any energy at all for water and salt balance. But the ionoregulators, that's a different story. Okay? Osmoregulators, they are not in balance. Okay? They have a different osmolarity than the medium in which they're found. Okay? And so this can be some submarine, mostly naphthol, primarily, and all terrestrial. And fresh water. Okay, freshwater organisms. They not only do they have a different osmolarity than the surrounding medium if they happen to be living in water, but they also have to regulate the ion. So these are always ionoregulators. Okay? Whereas this category, osmoregulator, ionoconformer, is contradictory. Doesn't exist. Okay, so that get it for you? Okay, yes.
Can you go for ionoregulator again?
Okay, an ionoregulator. They're isosmotic to the ocean water, meaning they have the same solute concentration, but not necessarily the same individual ion concentration. Okay, I gave the example before. This is inside. This is inside the animal. This is outside. And it might be in the ocean water. We might have sodium concentration, might be 0.4. Chlorine concentration might be 0.4 milliosmoles. And the magnesium might be, let's also say 0.2. All right, all that total solute concentration adds up to 1.0, right? Okay, well, it might be that inside, this here is 0.2, this is 0.2, and this is 0.6. It still adds up, well, no, that's right. It still adds up to 1.0, right? Total. So this is isosmotic. There's no net movement of water in or out. However, due to the fact that the solute concentrations are different, this tends to move in, this tends to move in, this tends to move out because of the gradients. And therefore, this has to continually use energy to pump, actually the other direction, pump this back out, pump that out, and pump this in. And so it is an ionoregulator. It's isosmotic, it doesn't have any problem with water moving in and out. But it has to continue, since it's maintaining a solute concentration for individual salts that are different than the medium around it, it has to regulate the ions. Okay, that's why it's called an ion regulator. That, get it?
Yeah.
Sorry, one last one. So could you give us an example between, or like, of an animal that has osmoconformer and ionoregulator?
That is an iono,
Regulator
Yeah.
that is osmoconformer?
yeah, a lot of crabs. A lot of crabs are this. Okay, and there's probably other crustaceans that happen to be like that as well. But I do know that some crabs, in fact, some crabs that species of crabs that live in the intertidal zone. Sometimes they can be terrestrial, sometimes they're in the ocean water, they're moving between two totally different environments, and they can switch between these two phases, okay? They have the physiology to be both, depending on circumstances.
Does that include Xanthosaurus, horseshoe crab?
Does it include what?
Xanthosaurus, horseshoe crab, or is it only crustacean?
Well, it's mostly just only these crabs.
Yeah, but you said crustacean and then you said crab. Do you also mean the horseshoe crab?
Oh, horseshoe crabs, that I don't know. I'd have to look it up. You know, because that I don't know for sure.
Crustaceans for sure. Okay.
Yeah, that I'm not sure about. But, you know, because there's various types of animals that belong to this, but when you ask for a specific example, crabs I do know, okay? But, and it isn't every species of crab. You know, some crabs that can switch between. Other groups of marine animals that are ionoregulators, I can't think of right off the top of my head, because I don't know which ones do what. I'd have to look it up. Okay? Okay, so continuing. This is where I left off. Just getting into excretion. Okay? What's important about excretion is it's getting rid of metabolic waste, and all these, and it's using the same organs for osmoregulation, pH balance, and getting rid of nitric waste.
Just to go back
Uh,
are we moving from the low to high?
Okay, the solutes always are going, since we've seen the solutes are going from high to low.
Oh, high.
Yeah, it's just like any sort of diffusion, okay? And in fact, when I'm talking about osmosis, the diffusion of water, we're still looking at the concentrations of the salts, the solutes, because it's easier to characterize those concentrations than it is with water. Okay, but nonetheless, with osmosis, that is the diffusion of water from an area of high water concentration to low water concentration. But we're looking at the solutes, okay? So where the solute concentration is high, the water concentration is low. And that's why it looks just the opposite, okay? Because the water will be moving from low solute concentration to high solute concentration.
So are the numbers there in terms of solute concentration or?
This is solute.
And then so would this move from inside for like, an
Okay, so if the sodium concentration is 0.4 milliosmoles out here and 0.2, okay, it's going to tend to leak in. It's going from high solute to low solute. Just simply because we're talking now about the diffusion of the solute,
Okay.
okay, not the diffusion of the water.
Okay.
Yes.
Are both happening at the same time, or no?
What's that?
Are they both happening at the same time?
Yeah, yeah. And in fact, to maintain balance, this usually takes active transport that back out, and it's happening continuously. Yeah, so that stays in an equilibrium. But it's a dynamic equilibrium, okay? All right, any other questions? Okay, now for terrestrial animals, the problem is water loss, okay, water loss. And therefore, they have to adjust for that. Now, the major terrestrial animals that are really terrestrial, that is, they don't have to live and breed in a watery environment happen to be the arthropods. Okay, they're truly terrestrial, and vertebrates. Okay, now when I say vertebrates, let's get more specific. It's the amniotes. Amniotes that are really highly adjusted to land. But for even earthworms, snails, they're still terrestrial. They usually stay in very damp areas, damp environments, but still they also, as terrestrial animals, have a water loss problem, all right? They sort of cut down the water loss by selecting a particular damp environment to live in. But as far as amniotes and arthropods, they have moved further away from water, more adjusted to land. But still, conservation of water is a problem. And so, in fact, it's so severe that if you're out and not drinking any water on a hot day, all you have to do is lose 12% of your water mass and you're dead. Okay? It's pretty fatal. If you remember the hiker family last year hiking near the Merced River that they found them dead, okay, both their dog, their baby, man, woman, and it was because, and for a long time they were looking for what killed them. What killed them, because it was a mystery that they all went together. But turned out that they lost too much water. It was a hot day, 103 degrees, somewhere around there, no shade, no water. They ran out of water. And later, when they were able to get their phones, get into their phones, they noticed, and this was in the newspapers just recently, even though this happened last summer, they didn't get the messages on their phones. They had been urgently sending text messages to friends, other people, telling their dire situation. But yeah, they lost too much water on a hot day. So keeping the water is real important. So what are the adaptations that have evolved in animals like this? For one thing, sense of thirst. Okay, you lose enough water, then your brain tells you that you're thirsty. You have that sensation of thirst that really drives you to go get something to drink. Furthermore, we have excretory organs, osmoregulatory, excretory, because these are doing the same thing, that conserve water. Okay. Now, to get rid of metabolic waste, you always have to lose water. There's no way around it, okay? Because the urea has to be dissolved in water in order to get rid of it. And so, but nonetheless, these organs are such that they can concentrate the urine so that we're not losing too much water. In fact, they're able to produce. Now, when I use this term, obviously, it's relative. Relative to what? Relative to the osmolarity in the interstitial fluids, or in the blood, okay? So here they can produce a urine that is highly concentrated compared to what the solute concentration is in the blood and in the interstitial fluids. So hyperosmotic urine. Frogs can't do that, incidentally. Frogs can't do that. That most they can produce hyposmotic urine or isosmotic urine, but they cannot produce hyperosmotic urine. But mammals and birds can. Another adaptation, of course, is to have covering that retards water loss. And that's having, with the arthropods, they produce wax. So on their cuticle, exoskeleton, they secrete wax, which prevents some of the water loss. And with amniotes, once again, change this to amniotes. Vertebrates is way too broad. Amniotes, more specifically, that they have layers of dead keratinized skin. That's different than a frog or fish, okay, because they don't have highly keratinized skin, but amniotes do. And this helps prevent water loss as well. Okay, so what types of osmoregulatory and excretory organs have evolved in animals? But in whatever case, I'm going to go through a few types. they all depend on a epithelium for transport. And this is for controlling water concentrations and salt concentrations at the cellular level. I mentioned it before, there are no pumps for water. How water is manipulated in the body is by manipulating the solutes. Okay, so there's pumps for solutes, active transport, and these create concentration gradients. And then the water follows the concentration gradient osmotically. And that's how things, all these epithelium happen to be working. So one type, primitive type, the earliest that we think that has evolved, some call it protonephridia. All these excretory organs are based on tubes, usually that consist of a simple cuboidal epithelium, most of the time. Simple cuboidal epithelium. And with the case of the protonephridia, the best example of where the protonephridia happen to be found is in flatworms, okay? So planaria, liver flukes, and cestodes, okay? They're examples of this. So this is a tube that has no opening, only has an opening to the outside of the body, okay? But it has no opening to the interstitial fluids. However, the tube is capped with a cell, special type of cell, that's called a flame bulb cell, okay? That's on the inside of the body. That is ciliated, and I'll show a picture here and explain it in a moment. But for flatworms, since they are living in an aqueous environment, okay, either planaria, they're in water. Liver flukes and cestodes, they're in your body, but they're still in an aqueous environment, okay? So as a consequence, this is only for osmoregulation, okay? Water can move into the body, okay, especially for planaria. They're living in fresh water, and therefore they have a higher solute concentration in their body than in fresh water. They always have water coming in, and therefore they have to get rid of it, all right? And so this is nitrogenous waste, ammonia. Okay, they don't have urea, they don't have uric acid. It produces just ammonia, and ammonia easily diffuses out of the body. They don't have to have any special type of excretory organ for that. So these tubes are all throughout the body, and they have an opening to the outside called the nephropore. That word should be up here somewhere. Or is it on the previous screen? No, it's right here. Anyway, that opening is called a nephropore. And that releases the urine to the outside. Inside protonephridia, it's capped with a flame bulb cell. And so this covers the end of the protonephridium. However, the plasma membrane here has sort of interdigitates with the other cells so that there's slits, real narrow slits there, okay? The cilia are on this side, only inside of the flame bulb cell that's inside the protonephridia, and that's what creates the force to draw in hemolymph. And so this are beating, and it draws in through these slits hemolymph that then has to be pushed down too. Since it can't escape up here, there's only one direction for that fluid to go, and that's down the protonephridial tube. 3 different processes take place as far as changing the hemolymph, which is now filtrate. Okay, once it gets into the protonephridium, it's called filtrate. But to turn it into urine that will exit through the nephropore, there's these 3 processes, and I'm not going to go into details about how these happen, reabsorption, secretion in a general, I will in a general way, but when we get into some of the other systems further down here, I'm going to go into much more details as to, at the molecular level, how these are achieved. So for one thing is filtration. Filtration is non-selective except for size, right? Except for size. So larger proteins that happen to be here in the hemolymph will not get into the protonephridium. But with the beating of the flagella by bulk movement, we have salts, glucose, amino acids, other things that happen to be small molecules dissolved we'll get into this too. And then as it goes down here, what's in the membrane of these, either the apical membrane or the basal membrane of these cells, are ATP-driven pumps for reabsorption. Okay? Now, what does that mean? What we have in here is glucose and amino acids. Do you think the worm, it's in the best interest of the worm to let that stuff go out with the urine? No. You want to keep that, all right? So these are pumped back out into the hemolymph. So that is reabsorption. Removing from the filtrate those things that you need, okay? There's also secretion. A lot of times this is for adjusting pH, okay, a lot of times. This is adding to the filtrate. So once again, that's in the wall of the cells that make up the protonephridium, transporters, a lot of times it's active transport, that if the hydrogen ion concentration, for example, is higher here in the hemolymph than what should be, then that is pumped across the epithelium into the filtrate. So that, in other words, this filtrate changes its composition until down here, okay? Because things have been added, things have been taken out. And what it then is released is the final urine, okay, out the nephropore. Now, this urine is going to be, since the problem with these worms is the absorption of water by osmosis, this is very dilute, okay? They try to keep most of the salts, most of and all the organic material, but it is very dilute, because mostly getting rid of water. Okay, and there's other animals that have protonephridia, but we don't need to worry about that. This is only an example I'm giving of protonephridia, yes?
Could you define the flame bulb cell?
What's that?
The flame bulb cell?
What about it?
Like, what is it?
I thought I already said that. Okay, that's all right, we'll back up. It's the cell that caps the protonephridium.
Okay.
And that has the cilia that beat, which create the force to bring in between these slits where plasma membranes from the tube cells interdigitate with the flame bulb cell. They have narrow slits between them. And so this is where the hemolymph enters. That's the flame bulb cell. It's called that because under the microscope, the movement of the cilia sort of looked like the movement of a flame on a candle. And that's why they came up with that particular name, flame bulb cell. Okay, does that get it? Okay. Metanephridia. Okay, this is what we find in olives, and also in some other animals, but this is the example I'm going to give you here. So earthworms. So even though earthworms are terrestrial, you don't find them in sand and dry soil. They're found in moist soil. And so there's water that are in the clay particles, and as a concentration, as a result of the higher solute concentration inside of the earthworm's body, water tends to move in, okay? So once again, it's a problem of getting rid of all this excess water. So with a metanephridium, they have two openings. An opening to the interstitial fluid, what's called a nephrostome, and an opening to the outside, which is also called a nephritiopore. I probably did that wrong. It's nephritiopore. Yeah, I left out two things on that, nephritiopore. All right, and there's similar types of situations here. Once again, ultrafiltration, reabsorption, and secretion. All those are happening in this particular tube. Okay, yeah, there's the word nephritiopore. That is where the urine exits the body. And so this is on the sides of, it has one of these on each side, left to right side, on every single segment. Every single segment. And a nephrostome and a metanephridium, a pair of them in every single segment. Okay? Every segment has one. Okay? So this is sort of a figure from your textbook, cross-section, nephrostome. Actually, so here's one segment And this is where most of the metanephridium happens to be. And remember that earthworms have a closed circulatory system. Okay, that's a little bit different than the flatworm, all right, that I talked about. A lot of this piping of the metanephridium surrounded by capillary bed. Okay, now the opening is actually not in the same segment. It's in the segment just in front of that segment. And so this is bringing in interstitial fluid. Yes.
It says hemolymph on the picture. Is it supposed to be hemolymph or
Does
not?
this say? Yeah. That's a good. I hadn't seen that before. This is wrong. Okay, that is not hemolymph. You don't have hemolymph in something that has a closed circulatory system. That should be interstitial fluid. Okay, that's a good catch. I hadn't seen that before. Yeah.
Is the name of the two openings the nephridiopore or
No, the nephridiopore is the opening to the outside. Nephrostome is the opening to the inside.
Okay, those are the two
Yeah, nephrostome and nephridiopore. Yes.
The green.
The green is the metanephridium.
Okay.
That's the metanephridium. And so it is taking in, and once again, so this has an opening, it's not little slits, it's a good opening. Ciliated, okay, because things don't just move in by magic. Fluids have to be moved by some sort of force. And so it's a beating of cilia that happen to be around the opening, the nephrostome, draws in interstitial fluid from the anterior segment, the one right in front of it. So it's not filtering the material in the major segment in which most of that metanephridium happens to be. So it goes into something called a bladder, and then it's propelled on through this highly coiled system, and we have the same processes of filtration. Filtration only in the sense that large proteins cannot get through that opening, okay? However, it is bigger than what you had with the flame bulb cells, and therefore still some somewhat larger entities, maybe disaccharides, small peptides, could still get in there, okay? But as it moves down through the metanephridium, we have active transport of materials back into, usually down here, into the interstitial fluid. So glucose, amino acids actively reabsorbed. And since they're right here next to this capillary bed, active transport moving higher concentrations of amino acids out. The amino acid concentration is higher than what's in the blood, and therefore we get diffusion back into the bloodstream. So this all goes back into the blood. And then secretion, usually of hydrogen ions, that's the case, pumped into it so that by the time we reach the nephridiopore, it is the mature finished urine. Once again, dilute. Once again, dilute. Okay, now pigment tubules. So we're going through all the things that you've seen in lab, right? Now with arthropods, insects, because this Malpighian tubules is in the insects, it's a blind end sac, but we still don't call that the protonephridium. It's not called the protonephridium because even though it has one opening, that opening is not to the outside. That opening is into the gut. Okay, so this was a transition between the foregut or the midgut and the hindgut. Okay, that's where all the Malpighian tubules happen to be connected. But the anus, the rectum, is also part of their system here. Okay, so this is in insects. And the driving force here is not cilia that brings material from the interstitial fluids, not the interstitial fluids, here it is hemolymph, okay, hemolymph. From hemolymph into the Malpighian tubule is due to an ATP-driven hydrogen pump that's in the apical membrane of these cells, which I'll show here in a moment. Now here, I'm going to go into a little bit more details about how these forces that draw in water and other materials happen to work. Okay, everyone have that before I go on. Everyone got it? Okay, so this is my drawing of Mount Picayune blind end, okay? Now what is in the apical, so this is the apical membrane, this is the basal membrane. This here is the hemolymph. This is the hemolymph, this is the lumen of the Malpighian tubule. So what is in the apical membrane is an ATP-driven pump for hydrogen ions. And so we're pumping hydrogen ions from the cell, from the cytoplasm of the cell, into the lumen. Now we're creating here an electrochemical gradient, okay? Because now we have high concentration of hydrogen ions here, but also we've moved a positive charge in here. So this side is positive, this side is negative. Now this is used, we have a co-transporter, which is an antiporter. Potassium, hydrogen antiporter, okay, or co-transporter. What's an antiporter?
Opposite direction.
Yeah, the two solids are going in opposite direction. Last semester, what I mostly gave you is symporters, examples of symporters. Even this semester, it was mostly symporters, but this here is a true antiporter. So that hydrogen ion concentration can be used in this co-transporter to go back down this concentration gradient, back into the cell, but at the same time, that energy is being used to pump potassium into the lumen. So green here is, when I have green arrows up here versus white arrows, the green arrows are the energy source. The green arrows are the energy source. This results in no change in charge, because we are pumping one positive charge back in to the cell, one positive charge out. And so they're balanced. We still have positive charge here because we've been pumping with the ATP synthase the hydrogen into here, but now it's that gradient that's used as for co-transport. What did that achieve? Well, yeah, what did this achieve? What do we have here now between the lumen and the cytoplasm of the cell?
Is there a concentration gradient?
What type of what?
Hydrogen?
Not of hydrogen,
Potassium.
potassium. We're lowering the potassium concentration here, high potassium there, low potassium here, but what about up here?
Low potassium.
Hi.
High potassium. We're pumping the potassium out. If this were originally in equilibrium, there was originally in equilibrium, by moving the potassium out over here, that makes this hot, okay? And so, potassium can be used as a gradient to be able to bring something else in. Now, also because we're putting charge here, hydrogen, and then later replacing the hydrogen with potassium, we're replacing the hydrogen with potassium. We still have positive charge here compared to this compartment, compared to that compartment. And therefore, to move chlorine into here, it just follows a charge gradient. Chlorine is negatively charged. So this is a chlorine channel, and this is a chlorine channel. And therefore, it is the positive charge of potassium that is leading to the chlorine moving in. Okay, everyone see that? But we also have another fancy co-transporter, this time on the basal membrane. And this is called the potassium sodium 2-chlorine co-transporter. It's a symporter, and it has a binding site for potassium, has a binding site for nitrogen, or not nitrogen, sorry, sodium, and binding sites for 2-chlorine. But what is the energy source that's going to drive that co-transporter? What?
Hydrogen?
ATP is for direct ATPases. This is a cotransporter. Cotransporters are driven by electrochemical gradients.
So the hydrogen gradient?
Where do you see hydrogen? This is, up there, no,
Will it also be by the potassium?
exactly. We've moved the potassium out, this is low, this is high. So potassium is a driving force. The green arrow is the driving force. So it's potassium electrochemical gradient. And notice, is this electrogenic? You remember that word electrogenic? A long time ago. Sometime December, maybe October. Any transport system that creates a charge difference on each side of the membrane is electrogenic. Is this cotransporter electrogenic?
Yeah.
Yeah.
Is it? How many positive charges here?
2.
How many negative charges?
2.
2. So, no charge change there. You're moving 2 positive and 2 negative, and therefore it's not making any difference in the charge across here. It's not electrogenic. This is. Okay, that is electrogenic. Okay, so potassium is what leads to the gradient here is what leads to the change in conformation. And once we have high, so notice here that chlorine has 2 different ways of getting into the filtrate. Okay, one is just by following the charge. Okay, but here we're increasing the concentration of chlorine by moving it from the hemolymph into the cell. We now have high chlorine, therefore out the apical side, we just require a channel. So a chlorine channel, because this is going down concentration gradient. And that's also true of the sodium. Because here, we're actively bringing it in through the cotransporter, high sodium concentration, and therefore you can just exit the apical side by simple diffusion, not simple diffusion, facilitated diffusion through a sodium channel. Okay? So we're bringing all this stuff in. What's going to happen to water? Because we're building up quite a concentration solutes here. We have a lot of potassium, we have a lot of chlorine, sodium.
It's going to be a low concentration also.
Okay, so which way?
Water's going to flow in.
It's going to go in. So we get water moving in. Okay, water moves in. It's following an osmotic gradient. And as that water comes in, some of these cells are a little bit leaky, and therefore there's a possibility of getting, a lot of times their tight junctions are, well, they don't have tight junctions, they put the protein junction, but sometimes they're not too tight. And so we can, with the flow of the water, bulk flow, get some of sugars, amino acids, and uric acid. Now, uric acid is very dilute, keep that in mind, because uric acid has very low solubility. But we don't want it to become a solid in the hemolymph. If it becomes a solid in the hemolymph, you can't get rid of it. And so the concentration is low enough that it is soluble. Okay? So that gets in here. Now, where's all this stuff going to go? Out where?
The tubule, so out of the,
We're talking about pig and tubules empty into.
the anus.
What do Malpighian tubules empty into?
With the rectum.
Not yet. Let's go back to a picture. Where did they empty?
Oh, the
Into the
Intestine?
gut. Okay, so all that stuff's going to go into the gut, all right? Okay, and why does it go into the gut? has to have a force. Why does it go into the gut?
Is it low pressure?
Why is there low pressure?
Because it's a higher volume?
Yeah, because we're moving all this stuff into it, right? There's no way out. So we move all this water into it, yeah, this is a higher pressure, it's gonna go down, flow that way. There's no other choice. So that empties into the gut. Okay, arrow showing that it goes that way. And, okay, now, in this area of the Malpighian tubules, we get reabsorption. Okay, because in here we have some amino acids, some glucose, vitamins, whatever vitamins they have. And those are things we don't want to get rid of. And therefore they are reabsorbed back in. And a lot of times hydrogen ion, if the balance pH is too high, it will be secreted into this area. So that what arrives down here into the gut is a lot of water and potassium, sodium, chlorine, okay? And that is mixed with the undigestible, unabsorbable material coming from the midgut. So here we have a lot of water and salts. They're important to this critter, and therefore we don't want to lose those. And so a lot of reabsorption takes place right here in the rectum. Okay? These cells are usually, it's an epithelium, of course, but it is a simple columnar epithelium. Much taller cell, but it's in the rectum, and there's a good reason why it evolved that way. So this material is rich in potassium, sodium, chlorine. Those are ions, electrolytes that are necessary for structure and osmolarity within the animal. We need to keep those, okay? And the water. So this is what happens in the rectum. Okay? All right. Now, I've drawn this not as a columnar cells, but they are, okay? So this is the lumen of the gut. This is the hemolymph, and this is the epithelium. And there's two driving forces here for reabsorption, okay? Two driving forces. We have two ATPases. One of these ATPases is a chlorine ATPase, which is in the apical membrane, and another, our good friend, sodium potassium ATPase. This is widespread in animals. It's very important ATPase. And once again, it's in the basal membrane. So these are ATP-driven pumps, requires ATP. So I'm not showing ATP here, but you have to understand that this is being phosphorylated, which leads to its conformational change, which brings the chlorine in, and then dephosphorylated for it to go back to its original conformation. And in the apical, well, first of all, if we are pumping in actively chlorine, we're building up a chlorine concentration here, high chlorine concentration. Therefore, across the basal membrane, we still have a chlorine gradient. It's high inside, low in the hemolymph. And therefore, all we need here is a chlorine channel. So that's a chlorine channel. It goes down its concentration gradient. Our sodium potassium ATPase is pumping sodium out, potassium in. But we have potassium channels in this apical membrane, so that this potassium can just go right back. Okay? Because what's happening to this potassium? We're building up a potassium gradient here too. But because we have this passive channel here for potassium, that potassium can just go right back, all right? But what we're building here, what are we doing across with the sodium? What's that going to do? We're pumping out sodium. That's primarily what we're achieving here. We're pumping sodium into the hemolymph. What type of gradient do we have across here for sodium?
This low sodium inside.
It's going to be low in here, but we have high out here. Okay, so the sodium can be simply reabsorbed through a channel. Okay, so this is a sodium channel, even though I made a strange symbol for it. Sodium channel. And then it gets pumped across the basal membrane back into the hemolymph. So the route here goes just like that, all right? So there we got sodium chloride. We moved sodium chloride across. What's going to happen with water? Yeah, why?
Because it's higher sodium.
Because of it follows the osmotic gradient. Therefore, water will move in, across, back into the hemolymph. What's going to happen to the concentration of material here?
It's gonna increase.
We still have potassium here and uric acid.
It'll be a high concentration.
Yeah, as we're taking out water, that's going to get more concentrated. And as you get more concentrated, then we have a good gradient for the potassium to move just through channels. So the potassium just moves through channels as a result of concentrating it due to moving the water across. And what's going to happen to uric acid as we lose water? Precipitates. It becomes a solid. And you don't want that solid to have happened way up here in the, right where the Malpighian tubules were. You'd plug up your digestive system. They're right here at the anus, where they can be pushed out.
Is that like the same as like kidney stones, kind of?
Kidney stones are not due to, well, sometimes they can be due to crystals of urine, but usually it's a problem with, because there's a bunch of pumps that I haven't talked about, okay, in kidneys. And if something's wrong with them and have a high concentration of calcium, for example, then you get precipitation of calcium, that's stones. And there's various types of stones. Sometimes it can be phosphate, okay? But yeah, but that's a concentration problem, similar. Okay? And therefore, our uric acid precipitates, and it goes out with the feces. It's now mixed with the feces right at the back door where it causes no problem. All right, so complicated little system, right? But so logical. The physiology of this is very, very logical. And we're going to see something a little bit different than this when we get now, our next stop is looking at mammalian kidneys.
Oh, can you explain like the potassium concentration in the lumen of the gut? Since it's high because the water goes in.
Which ion?
Potassium.
Potassium?
Yeah.
Okay.
Why does the potassium go to the hemolymph?
Okay, so first of all, coming from the Malpighian tubules, we put a bunch of potassium there. So the potassium, pretty rich here. But, and so if this is richer than out here, even without this pumping stuff, it will go this direction, okay? But if we had some sort of equilibrium here, because we do have this potassium being pumped in right here, which might oppose any of this coming in. But as we have moved chlorine and sodium over here, we put a higher concentration of solutes and the water moves. If you remove the water and the solutes can't go across, they become more concentrated, okay? Eventually that's going to reach a high enough concentration that you have a favorable gradient for the potassium to go.
And you said the potassium here is higher than the hemolymph?
Once you remove the water,
Oh, okay.
you know, as you start removing the water, this gets concentrated, okay? It's just like if you had sucrose and you mix it in with water, put it out in the sun, and let the water evaporate, what's going to happen to the sugar? In the glass of water. What's going to happen to the sugar? Does sugar evaporate? I don't think so. It's going to get more concentrated. Okay, so if you put a tablespoon of sucrose in 8 ounces of water and stir it up, and then put it out in the sun and let the water evaporate, the sugar is going to get more and more and more concentrated to the point it'll start to precipitate out. It's a good experiment. You have to do it something. Okay, does that make sense?
Yeah.
Okay. So, this system can adjust for calcium passively. Because if potassium, if it has too much potassium in its diet, in its hemolymph anyway, then this will remain, some of this will remain with the feces, and you get rid of it, because it has to be in equilibrium. Okay, but sodium and chlorine are really important. All right, now your kidneys, okay, you're the mammals we're talking about. All right, so this filters the blood, but once again, even though kidneys look like they're nice, solid objects, they're filled with tubes, okay, filled with tubes, epithelial tubes, and it filters the blood, okay, because you have urea in the blood that's produced from nitrogenous waste that are taken to the liver. So urea is produced by the liver, and it's at a concentration where it's not toxic to you. All right, so about with every cycle, approximately 1/4 to 1/5 of the circulation goes into the renal artery from the aorta, where the blood supply goes into capillary beds in the kidney, and it's a complicated process we'll talk about, the anatomy and everything. And so then that blood, after filtration, returns out of the kidney through the renal vein. And what would the renal vein go into? What's it connected to? The renal vein.
The inferior vena cava.
Right.
Vena cava, okay, inferior vena cava, okay? All right, so now, once we have this filtration, then there is a tube. We have two kidneys, left and right. You also have two tubes called ureter that go from the kidney to the urinary bladder. Urinary bladder is the lower part of the abdominal cavity. There's only one, okay, just one bladder. So, but it's taking in urine from the left and the right kidney. There is a sphincter right here, a muscle that holds this until stretch receptors in the bladder start to give you the urge to get rid of it, okay? So there's stretch receptors that can respond to stretch, and this sends a signal to the brain when those stretch receptors, when the bladder is full, that you have to, sometimes the urge can be really, really strong too. And then muscles will push this out into urethra to the outside world, and you get rid of it, okay? So let's look a little bit closer at the anatomy. Okay, this is, kidneys are set up here, okay, rather compact. However, they're filled with tubes and blood supply. So the renal artery and renal veins, they're not too long, okay, because these are lying real close to the aorta and the vena cava. Then there's the ureter coming down to the bladder, and then to the exterior is urethra. There's only one urethra. Now, the functional units inside of the kidney is something that are called, okay, so these are both for osmoregulation and excretion, but they are called nephrons. And so I'm going to go through the anatomy first here of a nephron, all right? And although this slides will show it, I prefer to draw it, okay? Because it's going to be a little bit more schematic, easier to see, all right? So a nephron inside the kidney is a tube system that, first of all, on the outside of the kidney, near the surface, it has what is called Bowman's capsule. And that is a simple squamous epithelium. Connected to this is a tube, that I'm just going to draw it like this, called the proximal tubule. Proximal tubule is one cell layer thick, it's an epithelium, but it is a cuboidal, simple cuboidal epithelium. Then this will make a hairpin turn, and I'll show that, when I say hairpin turn, I'll point it out a little bit later here on the slides. That goes in a different direction. And then deep inside the kidney, this makes a U-turn. And we have, this here is called the loop of Henle. Okay, the loop of Henle, because the tissue composition is bigger. What's that? Probably, yeah. And then this, so there is a descending and ascending loop of Henle. Okay? And then up here is now called the distal tubule. Okay, distal tubule. Now proximal, distal, proximal means it's close to a reference point. Distal means it's further away from the reference point. So what do you think the reference point is? Bowman's capsule. Okay, this is proximal Bowman's capsule, that's distal. And then this empties into, so this is one nephron. One nephron, the piping system, but not the blood supply, because I'm also going to show you the anatomy of the blood supply. All right, so that is associated with this. Several nephrons will then empty into what is called a collecting duct. Okay, and then this collecting duct leads to what's called pelvis, kidney pelvis. So it's not the same thing, all right? And a renal pelvis, right up there on the board, all right? Renal pelvis. So all of these up here are nephrons. Bowman's capsules are located near the outside. And in fact, this kidney has two different layers. It has an outer layer, which is called the cortex, and then the inner part, which is called the medulla. And so if I put the division here between cortex and medulla, it's not like that. This up here is the cortex, this is medulla. Okay? All right, blood supply, yes?
Sorry, what are the blue and red figures like the past few
On the previous one?
Yeah.
Yeah, these are, I'm not sure what they're trying to show there. Probably, these are Bowman's capsules. But why blue, why red? It's probably because there's two different types of nephrons. And they're Bowman's capsules of two different types of nephrons. The nephron that I just showed you is what's called a juxtamedullary nephron. This is what allows, now, reptiles such as lizards, snakes, and amphibians such as frogs and salamanders, they also have nephrons, but they don't have juxtamedullary nephrons. They have what are called just cortical nephrons. What's the difference between cortical and juxtamedullary? This here is showing a juxtamedullary nephron. And it has a long loop of Henle. So up here is the cortex. That's where Bowman's capsule happens to be, in the cortex. And of course, this is called, I'm just calling it the proximal tubule, distal tubule, but they're highly coiled, and therefore the actual name is proximal convoluted tubule, distal convoluted tubule. But we're just going to call it proximal and distal. That works just fine, okay? Because it's highly, goes around in the cortex. But the loop of Henle, if it's a cortical nephron, it really doesn't have a loop of Henle. So the material is just moving almost directly from proximal to distal tubule. And if there's a loop of Henle, it stays up in the cortex. But with juxtamedullary, they go way down into the medulla of the kidney. And this here is what allows, and this happens in both birds and mammals, this here is what allows concentration of urine. Okay, as I said, reptiles such as lizards and snakes, basically, and definitely frogs, they can at most make isosmotic urine. They can't make hyperosmotic. This is what allows mammals to do so, highly concentrated. Okay, and we'll see the physiology here momentarily. Okay, now the blood supply. I'm going to go ahead and use this for the blood supply. We have, of course, the renal artery. Then we have here a branch of the renal artery. So there's an artery. And that artery will flow into, in fact, this here, from that artery, the afferent arteriole. Okay, what does afferent mean? Okay, going in. Okay, so afferent arteriole, and it enters into Bowman's capsule. Okay, so really Bowman's capsule, if I were to draw this correctly up here. More like this. Okay, and I'm not going to talk about too much what's happening here because it's sort of complicated, all right? So this is epithelium, that's an epithelium, all right? And so we have this blood supply that's coming in. It enters into Bowman's capsule through the afferent arteriole, and then it breaks up into a capillary bed. But this is a very special type of capillary bed. And that capillary bed, of course, is inside Bowman's capsule, and that's called a glomerulus. Right there, glomerulus. So this is a capillary bed. And what can happen with capillary beds?
Exchange with
Exchange, okay? So what is real important here is blood pressure. Blood pressure, okay? Because this is under high pressure because it's pushing material out of the capillaries into Bowman's capsule. And then from Bowman's capsule, and this is by bulk flow, but being pushed by blood pressure, okay? So this filtrate will enter the proximal tubule. And we'll come back to that here in a moment. So this is, since it's bulk flow, it's ultrafiltration. Ultrafiltration. Now these capillaries are really special in glomerulus because they are especially leaky, okay, compared to other capillaries. However, what can come out into Bowman's capsule is basically plasma. And so large proteins, cells can't exit. Okay, it's only fluid, salts, small molecules. So urea and sodium, potassium, magnesium, calcium, amino acids, vitamins, monosaccharides, but not big things, okay? So filtrate, and that moves into proximal tubule. Now what about the blood supply? Exiting the glomerulus. So don't confuse the glomerulus and Bowman's capsule, because they're two different things. The glomerulus is a capillary bed, okay? So there is now an efferent arteriole, okay? And that breaks up into another second capillary bed, which your textbook, this is a photo from the textbook, and there's another mistake here. This up here, the capillary bed that surrounds the proximal and distal tubules is called peritubular capillaries. And so this is a separate capillary bed. This really should be called, instead of efferent arteriole, it would be logical to call it a efferent, well, it's a portal system. But still, the naming in the kidneys, since the blood supply is very complicated, we're not going to call that a portal vessel. Okay, it's going to be called the efferent arteriole, yes.
Are those capillaries for the absorption?
Are they what?
Are they for reabsorption or?
Yeah, we're getting there, exactly. You're looking ahead. Okay, you're right. Okay, and so, that's there. Then we have another capillary bed that goes around the loop of Henle. And this is called the vasa recta. Vasa recta. So we now, and what's interesting about the vasa recta is that it flows, the blood flow in the vasa recta is counter current to the flow of filtrate in the loop of Henle. This is the descending loop of Henle. Filtrate is going down into the medulla, and it's ascending, going back to the cortex on the ascending loop of Henle. But the blood flow in the vasa recta is going counter current to it. It's going down here, over to this side, going up. Okay, so the flow of filtrate and the flow of blood in both sides of the vasa recta compared to the loop of Henle, we have counter current. That's going to be significant, right? Then this will come into a short vein that will then go back into the renal vein eventually. So this is the flow of blood through the system. Okay? All right. Now, what's going to happen here? Filtration. We're looking at what happens in the urine. Okay, so we have filtration in glomerulus, which puts into the Bowman's capsule. Bowman's capsule, it basically is plasma, okay, very much like plasma. Now, there are, this filtrate still has to cross into the proximal tubule through epithelium. But I'm not gonna talk about this. This is a highly specialized structure that happens to be right here that allows this fluid to go in, okay? So, we're filtrating blood pressure, actually it's serum. I shouldn't have said plasma. Why is this serum instead of plasma?
Does it have no cell?
Yeah, so those thrombin and the prothrombin, all those blood clotting proteins, they're not gonna be in here. They're not gonna be in here. But this has glucose, it has a bunch of other things. Now, so we have filtration here. Contains these things, some nitrogenous waste, just small molecules. Small molecules. And then we get secretion, can happen, and that's usually active transport into the tubule, it's adding to the filtrate, but we also, and this secretion can happen in the proximal and the distal tubules. Okay, it happens in both places. And this is adding to the filtrate. But we have a bunch of stuff in this filtrate that we want to keep. Now, this can be done, this secretion, a lot of times active transport, okay, because they're on both ends, but especially in the proximal tubule, there is a hydrogen ion ATPase, okay, that pumps in, and it's in the apical membrane of these cells. But due to gradients, we can also have passive, that is facilitated transport, going down gradients, depending how low the gradients are produced. But we also get reabsorption. Reabsorption is real important here. So it reclaims these essential small molecules. And reabsorption reclaims water. We have a lot of water that's going into all these, I'm just showing one nephron, but we have thousands of them, okay? And reabsorption of water can happen, and also of solutes, and I'll specify this in a moment. Proximal, distal, loop of Henle, and on the descending loop of Henle versus the ascending loop of Henle, conditions are different. That epithelium happens to be different with regard to what types of transporters happen to be in. And also the collecting duct. And so there's many places to adjust the solute concentration, water concentration, and so forth as we go through this. Now it's the proximal tubule is where all the organic material is reabsorbed. proximal tubule only, okay? By the time the urine or the filtrate reaches the loop of Henle, there should not be any glucose there. There should not be any well, that could have vitamins, because if you take too many, you'll overwhelm the system. But no glucose or amino acids, fatty acids, short chain fatty acids should not be there. And how is that achieved? Okay, so if we have here we have capillary, paratubular capillary, okay, this is the lumen of the nephron. This is the proximal tubule. Can you guess what that would be?
Glucose transport?
Sodium potassium ATPase. And so it's pumping, driven by ATP, sodium out to potassium in. So what's it doing to sodium concentration on the apical membrane? This is the basal membrane. This is the basal, this is the apical. The apical is always facing the business part of what we're trying to do. We're trying to be absorbed. So what's happening to the sodium concentration here?
It's increasing. Or,
You have to tell me which compartment. Is there a gradient?
well, yes. Because the inside is low concentration now.
It's going to be low in here, high here. Exactly. Because first of all, when we were in, up here in the glomerulus, since this is bulk flow, initially, the sodium concentration here is the same as in the blood, the same as in the interstitial fluids. They're all in balance initially. But now we're pumping it out. Okay? So that's going to lower the concentration of sodium here. So this is going to be low because we're pumping it out. But what's the sodium concentration here? It's going to be high. And what's concentration in the blood? It's low. Therefore, to get it into the blood, you just need sodium channels. It just flows by sodium channels. So we're reabsorbing the sodium, okay? And what's happening with chlorine? What is happening to the chlorine?
Is it following like just the charge gradient?
Because it has negative charge. We're moving positive charge that way, just through channels. Facilitated transport just through channels. It's following the charge, okay? Now how would you design this system to get our glucose back? We have no glucose ATPases. There's no such thing. So how do we get our glucose back?
GLUT2?
Well, that's passive transport. How do we get glucose back in? It's part of the story.
Is it the
It is a, what? Say that again? Sodium gradient. We created a sodium gradient, right? We're pumping the sodium out. That's high sodium, okay, down here compared to low sodium in here. You put a co-transporter. So you have a sodium, glucose, co-transporter. What's the driving force? Sodium. Sodium, because it's going to be pumped out, and glucose is carried along, okay? So glucose is co-transported into the cell, and now that we have high glucose, compared to up here, because first of all, this glucose and that initially were at the same concentration. But now we're moving glucose into here, it's high, here's where you can have a GLUT transporter. I don't know if that's GLUT2, because we have about 12 different types of glucose transporters, okay? But nonetheless, it's a glucose transporter. You have a gradient, and we get it back into the blood. And that's also true of, let's say, amino acids. Let's say valine. Then there is a sodium valine cotransporter. Once again, the driving force is the sodium. And that will bring our, and so all these things, some of the vitamins, it depends on their concentration, they're brought back in by sodium gradient and cotransporters on the apical membrane and then facilitated transporter on the basal membrane. Yes.
Is the co-transporter specific for valine only?
It may be for nonpolar, several nonpolar, you know, because things can be, some of these transporters are conserved. Well, evolution is chosen for economy, okay? But still, a lot of the chemical natures of these R groups are sometimes quite different. It's possible that the same co-transporter for lysine and arginine, because lysine and arginine have very similar R groups. Valine, alanine are also similar as well. So that is a possibility, okay, but I don't know specifically, but we still would have more than one type of amino acid transport, co-transport. That, and also for some of the monosaccharides, mannose and other things, there would be different co-transporters. I know that like in the gut, the glucose co-transporter, sodium glucose co-transporter is not the same thing, would not carry fructose, for example. Fructose has its own transporter. Okay, so all this is moving here. Secretion. Here we have a hydrogen ion. And this here is in the apical membrane. That would transport, depending, these transporters here ATP-driven transporters, they can be shut down, inhibited, or activated due to complicated systems that we don't need to go into. So if we have too much hydrogen ion, the pH is too low, then we get secretion. And then out here, of course, we have hydrogen and interstitial fluids. It can be brought in through a hydrogen ion channel. Okay, so it's in the proximal tubule. Now, what else here? What else would be leaving? What else could be reabsorbed back into the blood?
Sodium?
Well, we already have sodium here.
Potassium?
Okay, something real important. There's a lot of water that goes into this system. We need to get that water back. Water, okay? How is it moving? Or why? Why would water go up here back into the blood? Because all this sodium, chlorine, amino acids, sugars that we've reabsorbed, those are solutes. And so that's building a high concentration up here, and the water follows osmotically. Now, what's in both of these membranes are aquaporins. Okay, those are channels for water. All right, so these have a lot of aquaporins. All right, so all the organic stuff is reabsorbed here. If an individual ends up with glucose in the urine, that's a real good sign that the individual has diabetes. In fact, that's one of the major signs of diabetes that can be caught very early on, is high, because there's so much glucose in our blood, because that's what diabetes is, high glucose levels, that it overwhelms the system to be able to take it out. And therefore it ends up in the urine. Okay, all right. So now we have just urea, still water, we still have a lot of water. Okay, so all this is incidentally, how much blood goes into your kidneys per day is about 1,000 to 2,000 liters. 1,000 to 2,000 liters. Okay, so your blood, you have usually about 5 liters of blood, right? Obviously, you're not losing all the water in your blood. And you're not urinating that much out either. At least, we hope not. You'd be dead, okay? So, all that fluid is going through the kidney tubular system. About 180 liters ends up as filtrate in the proximal tubule, okay? But you don't urinate 180 liters a day. So it's about 1.5 liters, right? There's no way to get rid of excess sodium, excess chlorine, urea, no way to get rid of it without losing water. You have to lose some water. There's no way around. But depending on the environmental situation, you may want this to be very, very concentrated, to lose as little water as possible, unless you've been drinking a lot of water. Then you want to get rid of a bunch of water, okay? So the kidneys are really remarkable in how they can adjust. They can produce a very concentrated urine. They can also produce an not isosmotic, but hyposmotic, a very dilute urine compared to the blood. So so all that, most of it is returned back to your blood, the water. Okay? So only birds and mammals, and the reason this can be hyperosmotic is because of juxtamedullary loop of Henle. Okay? Juxtamedullary nephrons. Okay, now, what happens as we go, so all these are showing active transport, secretion, and reabsorption. Of course, all this stuff that's coming out of the proximal tubule will go right back into the capillary beds, okay, the peritubular, because the peritubular capillary beds are right next to these tubes. But now as we, and so the concentration of solutes in your blood is 300 milliosmoles. Okay, that's, for humans, it's about 300 milliosmoles. So this filtrate, as it goes into the loop of Henle, is initially 300 milliosmoles. But the plasma membranes of these loop of Henle cells going down are impermeable to salt, but permeable to water. Now, in the interstitial fluids, there is a salt gradient from the cortex to the medulla, and that's what's shown over here. Up here in the cortex, it's isosmotic with the salt concentration within the interstitial fluids is isosmotic to the blood, 300 milliosmoles. But we have a salt gradient, a concentration gradient, as you go into the medulla, that in humans can go as high as 1,200 milliosmoles. The plasma membranes of these cells on the descending loop of Henle have a lot of aquaporins, so they're permeable to water, but they're not permeable to the salts. There's still salt in here. There's still sodium chloride. There's still magnesium and other ions. It's still there. So as we move down this gradient, right here we have, first of all, 300 milliosmoles inside, but in the interstitial fluid it's 500. What happens? This is what we have. Put it over here. Inside loop of Henle, this here is 300 milliosmoles. Out here is 500.
Don't move the inside?
Think again. We're talking about what can't move is the salts. Solutes can't leave.
Oh, so the water will go
Water
back.
can leave. We have, as I said, there's a lot of aquaporins there. So which way is water going to go?
To the 500
It's going to move out. These are solute concentrations. Okay? Osmosis is from low solute, the movement of water from low solute to high solute. Okay? So this moves out. And therefore, this concentration, now the salts can't move out. The salts have to stay there. So if we're moving out water, what's going to happen to the solute concentration?
It's going to increase.
It's going to increase till it is in equilibrium. But this is moving this way. Now we're down here where it's 500, but now it's 700 out here. What's going to happen?
Osmosis will continue.
Water's going to move out until that becomes 700. Concentrated, but it's moving this way. Now it's down here, 700, where outside is 900. It's going to move out. So the solute concentration, because we're taking out water, and where does that water go? Into the vasa recta. Okay, it's being reabsorbed. It goes back into the vasa recta. And I mentioned that the flow of blood in the vasa recta is countercurrent. That's going to be significant. I'm not going to get to it today. Therefore, you don't have to worry about it on next week's exam. But I'll get to it later. But nonetheless, point being here, we're taking out water, the salt is becoming concentrated. We turn the corner, and now we can get this as concentrated as 1,200, hyperosmotic. But now as it turns the corner going up this way, the membrane is a little bit different. It has no aquaports. Water cannot move out. Water can't move in. There's no aquaports. But it has sodium channels. All right? So here we have 1,200, and we've moved up to this position, which is now 900. What's going to happen? Okay, let me go back over here. Your kidneys are so beautiful. I love kidneys, okay? So now we've concentrated that now to 1,200. On the ascending, no aquaporins. No aquaporins, water can't leave. But we have sodium channels. So now we have here 1,200 solute concentration, but out here it's 900. Is anything going to move? Is anything going to move? Water can't move. But we have sodium and chlorine channels. This is the sodium chlorine concentrations. Is there a gradient? Which way? So, yeah, so sodium is going to leave. And chlorine follows it because of charge. What's that going to do to the concentration inside the tube? We're removing salt. Is it going to increase? We're removing salt, but no water. Taking the salt out, but not the water.
It's a low solute content.
It's going to go lower, okay? It's going to go down to where it's in equilibrium. Now, an interesting thing happens right here is active transport of sodium out. This requires ATP, okay? So it's a sodium pump. In fact, it's related to sodium potassium ATPase. We're not going to go into that details here, but the chlorine, of course, follows the charge. And so, first of all, we have up here, concentration is 900, but it's 700 out here. We're actively, in fact, we can get way out of balance here. We put pumping the sodium out, we're going down, making it less and less dilute. So this up here is active transport. And right here, the blue. So as we're going up here, water can't leave. We're removing the salt. And therefore, the concentration gets back down here to 300 milliosmoles. And up in the distal tubule, this is a bunch of pumps, ATP-driven pumps up here, and also co-transporters. This is where other ions, such as potassium, bicarbonate, magnesium, calcium, etc., this is where they're adjusted. They can be reabsorbed up here. And this isn't showing the ATP pumps that are necessary for reabsorbing calcium and so forth. But as we're doing that, removing sodium and so forth, that we can make hyposmotic, 150. The interstitial fluids are 300. If you drank a lot of water, this is what's going to happen when you urinate it out. It's going to be hyposmotic. We've got to get rid of all that water, okay? But there's other things that happen as we go down the collecting duct, okay? And I don't have time, fortunate for you. So this will not be on the exam. The loop of Henle here, those relationships will be, okay? Because this is where the cutoff for the exam is going to be. Okay, yeah.
The liquid that's inside the tubule is that like serum that's being filtered?
Well, it will be when we get down here.
So it's still serum down there?
Yeah, the final urine is produced in the collecting duct. Okay, because see, we've taken out, first of all, this was serum. Now, and it's being modified as we go through the system. First of all, we take out all the organic stuff. And so we have just only urea. Now, what do you think?